Ed's planning to come.
Robert?
Hello.
Bob and I met, how long ago? Was 10, 12 years?
About 10 or 12 years, exactly. I saw the light 10 or 12 years ago, thanks to Robert Hester, because I didn't know anything about much of anything. And then I heard about HUMOD and the incredible vision and technology that Robert is the caretaker and developer of, and it just became one of the big inspirations of my life from that point onward.
Oh, you're too kind, Bob.
Oh no, I'm just telling the truth.
Well, I know that was one of your first suggestions, Bob, that I get in touch with Robert. I'll mention too that I had the privilege of being escorted out of a Mayo Clinic physiologist's office when I asked if he wanted to get involved in leading some of our system dynamics biomedical applications. And he said, you need to talk with Robert Hester. So I did.
I didn't know I was that well known.
Oh, yeah. I think what I'll do is, because we're at a couple minutes after the hour, we'll go ahead and get started. I expect others will join. Coming on, I appreciate each one of you showing up on time. I appreciate that. So Robert is the current director of the Center for Computational Medicine at the University of Mississippi Medical Center. He is a Billy Guyton Distinguished Professor there. And for 40 years, and he's been working with integrative physiology in the last 15 years, leading up extensions to HUMOD, using computational physiology, computational medicine and physiology. And he has generously shared with me some things he could do. He was kind enough to respond, say, sure, he'd come and give us a talk, kind of explain this thing. I should mention he's the CEO and president and CEO of HC Simulation LLC, which is that's a commercial software development company looking at commercializing HUMOD. So he put a talk together today entitled Virtual Digital Twins. He's going to go through that. We can maybe take 35, 40 minutes is what we mentioned, whatever time it takes to present, have some Q&A afterwards. If we do run out of time, you can just, and you have additional questions, you can either email me and I'll follow up with you on that, or you can just enter it in the chat.
Yeah.
Robert, we really appreciate you making time to be with us today.
No problem. Yeah, it was, Jim contacted me and we actually met in Minneapolis last week. I was at the Design for Medical Devices meeting. And it's very interesting. I got asked by a friend of mine, Mark Horner at ANSYS, to be on a panel on Wednesday. And I got up and, and somebody said, well, when Mark introduced me, he said, well, we have somebody here from Medtronic who's the gorilla in the room. And then you have somebody from HC Simulation. And I got up and said, yeah, I'm the smallest one. I got 1.5 employees. But hopefully we can show you what we ended up doing. So there's my email addresses, and we're going to talk a little bit about developing digital twins. There's some acknowledgements to give forth right off the bat. Tom Coleman, his name is going to come up in a little bit. He basically started this project back in the late 60s as a graduate student. He sadly passed away last year. Drew Pruitt and John Clemmer both joined Jim and I for dinner last week, and Tom Lohmeyer was involved, retired professor now, did some work on this, which we'll bring up in a little bit. So I have no idea what your backgrounds are. I am trained Ph.D. in biomedical engineering, but really all that was done was I'm an integrative physiologist. So I'm gonna give you a little definitions about what we're doing. And basically physiology is the science of the functions of living organisms and their parts. I think and their parts is critical. So when we try to model physiology, we want to allow one variable to change while carefully controlling every other thing in an attempt to understand how A affects the system. So we talk about physiology. This is a very simple physiology. Some factor causes a response. You give insulin, glucose goes down. Very simple thing what happens from that physiology standpoint. Or you can have three things that can affect the response in terms of whatever the glucose response could be insulin. It could be that bagel you had in the morning, or it could be exercise. All these things can happen. But how this sort of gets a little bit more closer to what we consider integrated physiology is that factors can affect other factors who can end up having some effect on the response. And this is sort of very simplistic in terms of what we're doing with this. So, so we try to replicate something through something else. So it's usually generally simpler. It has a purpose or a set of purposes. And we want to sort of accomplish one and two simultaneously. So this was the original Guyton model in 1972. Excuse me, let me say Guyton Coleman. Tom Coleman was a Ph.D. student, electrical engineering undergraduate degree, master's. Came to Jackson. He did some animal studies and stayed on at the faculty. But this was the original model that was done, again, published in 1972. If you look at it carefully, you've noticed it's an analog model. It was not a digital model, digital computers, because those were not at the level to do this. This one had about 350 variables and parameters. And Tom's and Dr. Guyton's whole goal was try to understand what causes high blood pressure. And basically, what the model predicts and what the results over the years have really demonstrated is that it really comes down to the kidney. So basically, you just think about, you have a closed system and you take in water, what's going to happen to the pressure inside that closed system? It's going to go up. And if you take out water, it's bleeding, it's going to go down. And there are lots of things that affect the control of that. And if you think about it, most antihypertensives if you're taking that, have to do something with the kidney to excrete one mechanism or another to excrete more salt and water. So this is the current version, one of the versions. We actually have, this one has about 10,000 variables and parameters. We actually have one we've developed for medical training for the military, which actually has about 40,000. It's a little bit more anatomically correct. It has arms and legs. This one doesn't. This one just has skeletal muscle, which works fine for what we want to do. So this is where we really started off, and we do this with our graduate students right now. In terms of integrative model, it's basically a lot of feedback loops. Very simple in terms of this, looking at heart strength affecting a curve, Frank-Starling relationship. If you have an input of right atrial pressure, you get an output of cardiac output, and some other things with here, blood volume and things like that. And what we end up doing is connecting loops and influence each other one. So we start off with a very simple, and that's what we're actually doing with our second year students right now. They're doing a simulation project, and they start off very simple with this and start connecting all sorts of loops. And the whole idea is we want to have a stable dynamical system that evolves over time. I'll show you in a moment toward the end a simulation that basically lasts 3 years in the life of a person. So they need to be robust. They need to have all sorts of perturbations and respond to them. We have multiple systems. That means more ability to interact with the model. So we have pharmaceuticals. We can exercise the patient, the person. We can put them at altitude. I like to do that for my medical students and sort of look to see what the responses of going to the top of Mount Everest are. We can provide injury. Some of the stuff for the Department of Defense, we're doing it, whether it's a burn, pneumothorax, air blockade, a hemorrhage, or things like that. And basically, we go with one model to really look at multiple hypotheses or theories to be tested. Limitations, they depend on a whole lot of assumptions, which we hope we make the right assumptions. And again, while they're large, they do not contain everything that might be of interest. One of the things, a couple of things we do not have in HUMOD, mainly because we don't have the expertise and haven't had anybody looking at that, is inflammation. Inflammation is really a big deal, and a lot of inflammatory markers that have effects at different tissues. And it's just one of those things, as a small group of people working on this, it's challenging to add that into it. We do have scaling. We can scale body size. We can have a female version. We can have a male version. The one issue with the female version is we do have sex hormones, progesterone and estrogen. We do have a model of the menstrual cycle in terms of the hormonal effects with that. The challenge we've got is, how do those hormones affect different physiological pathways? And that is really a problem because experiments are not done to really test that. I have some colleagues that look at effects of estrogen and progesterone on different pathways, and they use animals, and what they'll do is ovariectomize them and then give them a pellet so it releases a constant dose of estrogen or progesterone, which is really not what happens in females. So there's some challenges that are having with that in terms of adding models, just because we don't have really good data or experiments that are not able to be performed and good data that's collected with that. Okay, so that gives you a little bit more information about the model. It is deterministic. It's all mathematical equations, about 8,000 variables and 2,000 parameters. It has 14 organ systems, two-sided heart, lungs, left and right kidney, which are also different sizes. One of them is a little bit larger than the other, as in normal people. Liver, skin, skeletal, respiratory muscles, GI tract, bone, adipose, and brain. It has a full endocrine system. The model is really designed for a cardiovascular renal system, to study cardiovascular renal effects, and pretty much everything you have there. A lot of that is cardiovascular renal, but it does have insulin glucagon and some other hormones with that. Full nervous system, reflexes, chemoreceptors, and afferent and efferent nerves are built into the model. So I'm going to give you a little study as what we started using the model for a couple of years ago with Tom Lohmeyer, who I mentioned earlier. So Tom was working for a company called CVRX, had a device to try to lower blood pressure. And you sort of wonder about, why do you have a device to lower blood pressure when there are all sorts of medications? Well, my wife is actually an internist that specializes in high blood pressure. And the challenge she always has is patients either don't have access to their drugs or they don't take them. So if you have a device that's working 24 hours a day, 7 days a week, then therefore you don't have to worry about that. So the basic idea here is, and I'm gonna at least show it on my, there are actually some nerve endings up in your carotid body. And let me back up. I have no idea about anybody's knowledge base. So I'm going to be fairly basic in a couple of points right here, particularly in terms of physiology. So you have some carotid arteries up in your neck, and around those arteries are some nerve endings. And basically, this is what prevents you from fainting when you stand up. So basically, if you go from a lying down to standing up position, what happens with gravity? It wants to pull all the blood to your feet, and therefore you get lightheaded. Well, as soon as the pressure falls, those carotids collapse, they get smaller, and the nerve signals are inhibited. The nerve signals going to the brain are actually minimized. So what happens is it causes your body to constrict the blood vessels in your legs, to therefore you increase the pressure and get blood to your brain. This is a little bit different one. What we want to do is stimulate the well, this is the standing up part. We decrease the baroreceptors. What happens is you increase, it goes through the brain, it's all integrated. It increases your nerves to your muscles and to your kidneys, and it actually ends up causing a decrease in excretion of salt and water. And the net effect is you bring your blood pressure back up. That's sort of a long-term goal. It also does the converse on a short-term goal when you're standing up. But this is the whole idea, was that for people with high blood pressure, we need to lower the blood pressure. So what happens if we stimulate the arterial baroreceptors? And so you get the arrows going the opposite direction. So this is what the device looked like. They went from 2 leads to 1 lead, and basically it's a pacemaker that pulses the signals on the carotids that are in your neck. So what happens if you stimulate the baroreceptors? As we said, you decrease your renal sympathetic nerve activity. What happens is your renal excretion goes up because you've decreased that ability to retain salt and water. And the net effect is you would lower your blood pressure through a variety of reasons. Turns out, very good idea, physiologically a very good idea. So Tom Lohmeyer was working with CVRX, and he used a series of animals, and this is what he got. And this is a paper we published back 12 years ago. So the red bars are actually in large animals, and these are daily averages. The black line is actually the simulation. So what you actually see is, as soon as in the animals in which they turn the stimulator on, there's a fall in blood pressure, and it stayed down as long as the stimulator was on. You turn it off, and it actually comes back up as on the right side of the top graph. Heart rate goes down and stays down. Sodium excretion, it's a little bit not the same, and really because he was doing daily averages, and we were doing basically every 1 second simulation, but it's relatively similar. So you can see the model did a very good match the experimental data very well for that. So Tom Lohmeyer's, oh, this is great, this is really happy, and it sort of showed what was going on. Well, the whole basis of this is, let me go back, is this renal sympathetic nerve activity. These nerves go into the kidney. So what happens if you remove the nerves to the kidney? Are you still going to get the same response, or are you going to get a response, no fall in blood pressure? Because it's all, the theory is based on you decrease the sympathetics to the kidney. So what happens if you block this right here? So the idea is if you stimulate that, there should be nothing happen because the pathway is completely gone. So Tom Lohmeyer actually went, and that was what he did in a series of animals before he did renal denervation. And lo and behold, when he did the renal denervation, he got exactly the same response. So he was sort of freaked out because he was working for this company, and it was, the whole idea was this happened through renal denervation, and it turns out the device worked, but you didn't understand why it worked. It wasn't the way everybody thought it would work. So that was an interesting point. So John Clemmer went and did some experiments. And the couple of things nice about the model is you can set things so they never change. So whatever the input is doesn't matter. So here's the basic thing that happened. This was published in 2018. Upper left is mean arterial pressure. You turn the stimulator on, pressure goes down, falls and stays down. Heart rate goes down and falls down and stays down. But what's interesting in the upper right is a hormone called atrial natriuretic peptide.
Are you talking to me? I'm in a meeting right now. I didn't finish this, I'm sorry. I didn't close the word.
So, okay. So A&P.
Jim, you can mute him. Jim.
Yeah, make sure you muted everyone.
Okay. We
You
back?
can mute him too. Yep.
Wayne, I think you might need to mute your mic.
Yeah.
Okay, we're going. So atrial natriuretic peptide is a hormone that's released from the right atrium of the heart. So remember, blood comes back from the circulation to the right atrium, goes into the right ventricle pumps the blood through the lungs, it goes back to the left atrium and left ventricle. So what happens is if you get too much fluid volume in your ventricles, it suggests your body has too much fluid, and so therefore it releases a hormone which causes you to excrete salt and water. So that's the whole purpose of A&P, is to cause you to excrete salt and water. It makes a very good antihypertensive if you could develop a drug, an A&P drug, but has not been feasible over many years of people trying to do it. So what you see is basically, in this case, the reason you have an elevation in atrial natriuretic peptide is the heart rate slows down. because when you get rid of the sympathetics, it also is what causes your heart rate to go up. So therefore, if you decrease your sympathetics, your heart rate's going to come down. So the heart rate goes down, it causes an increase in the H1 natriuretic peptide. So what happens if we set the heart rate so it doesn't change? Nothing is going to happen, it's not going to change. And what you see is A&P, you still get a slight fall in blood pressure, but A&P does not change. And so therefore you do not get the response that you would expect. So the device works as designed, but it doesn't to lower blood pressure, but it doesn't end up working at it through sympathetic nerves. And in fact, what happens is it causes an increase in A&P, which causes an increase in urinary output, which then causes a decrease in blood pressure. So we've done there's another device that's out there called AV Fistula, which is in clinical trials. And basically it works the same way, but I won't get into that. We've also published a paper on that also. So there's a couple of things we've done with it. Okay, so now let me get to the virtual population. And this is where things get really sort of interesting. When we started doing this model, we looked at it, it's because you could get the same response every time you did something. And it's sort of boring. I hate to say it, because we're not all alike, you know. And so therefore we decided, let's see how we go about creating virtual populations. So basically we wanted to test the hypothesis that we could simulate a virtual population, which was comparable to an experimental population. I got a little grief about this the other day, and I'll tell you about it in just a moment with that. So what we end up doing is basically, you look at the top box and there's a bunch of curves up there. And this is basically all the things that affect one particular set of blood vessels in the kidney. So blood comes in from the renal artery. It goes through what's called an afferent arteriole before the glomerulus, which is where everything is filtered. And then there's an efferent arteriole. So again, these are three things. There's some pressures, there's some A&P, there's some other neural activity, and there's this is basically a drug effect on the far box on the right. So what we end up doing is have a thing that affects blood flow called conductance, which is a combination of each of these. But then what we end up doing is we can actually shift the curve to the right or left, depending on how we wish to change the parameters. So that was the first thing we do, and it's really gotten quite interesting in terms of doing that. I'll say this right now, we can end up creating very large data sets that are dynamic, and I'm gonna give you an image, tease you a little bit with an image at the end about some idea about what we can do. So one of the things we did was a little bit different clinical sort of virtual clinical trial. There is a device out there. Remember I said the baroreceptor stimulation worked through renal sympathetic nerves. So in this particular case, what they were doing was going through and removing the renal sympathetic nerves. Forget the baroreceptor stimulation, just surgically go in, and it was called a renal ablation, and they would just use electromagnetic wave to destroy the nerves. Run a catheter up your femoral artery into the renal artery, and you could end up basically frying their nerves. And so we created a virtual population, 1,000 virtual hypertensive patients with a mean pressure of 100, not very hypertensive, but they were hypertensive. What we then ended up doing was basically removing the sympathetic nerves and looked to see what their change in blood pressure was. And we got something like this, which is the light colored and the shift to the left. So in this case, what you see is there is a significant group that had a lowering of blood pressure. However, notice what's on the right is you actually had a few in which blood pressure went up. Now, it's really not what you want to do, is to do a treatment and make the matters worse. And when you look at it in terms of basically that group on the right and pull it out in terms of change in blood pressure, you can see as, yes, we had this group on the left that had a 20 to 40 millimeter mercury fall on blood pressure, 20 to 30 millimeter mercury fall on blood pressure, sorry. But we have these that basically had less than a 10 millimeter mercury, or actually end up having up to a 20 millimeter mercury increase in blood pressure. Not really what you want. So
Scott,
we
did you have a question? Osgood, did you have a question?
I'm sorry.
Yeah, may I- Mhm.
Yeah, I'm not looking at the questions, but go ahead. If you got a question.
Sure. It's very quick. Can you please go to the previous slide?
Sure.
Or, oh, okay. So this distribution for the mean blood pressure, so for a normal population, there is a distribution for the mean blood pressure, right? Is this how it looks like, or is this something that you
No,
You
yeah,
created?
let me go back to the previous. So
Exactly,
normally,
this one,
so normally you have a distribution. What you would like to have is a normal
Mhm.
distribution where the mean is right about 100.
But does it look like normal distribution? I mean, I did
it-
something similar for the PSA, like values for human males. For example, it looks log normal. So is this how it looks like normally for mean blood pressure? I'm asking
It-
that because I know it. I don't know.
No, it's to be honest with you, that's what you would theoretically want, but the problem is it's probably shifted. It's probably got a more tail
Mm.
nowadays on the right-hand side. And
Yeah.
again, it's looking at, sure, if you have everybody treated properly and they're taking care of themselves, hopefully you would have a normal distribution. But we don't have a normal because people don't take care of themselves. People don't take their drugs. They've got a lot of other comorbidities. But you'd have to go back and look at large data sets. But the problem is that the data sets, you have to know what their underlying pathologies are or their underlying conditions are to really pull out, okay, this person has normal blood glucose, they have active normal kidneys and everything else. So I think in most cases, if you looked at animals, animals would have a normal distribution because you can put those in a controlled environment that have they're non-hypertensive and look at their blood pressure, and you would have a fairly tight normal distribution.
Mhm. I think one reason this might be important is there are recent discussions about the definition of hypertension, right? In the US, yeah.
Correct. So usually the idea was you want to get your systolic below 140. Now they've actually dropped it down, I think, to 130.
130, and now some people even argue that it should be decreased to 120. I mean, in some journals.
In some journals.
Okay.
Yeah.
Exactly.
The challenge is even getting them to 130 is because either, and we can discuss it in a moment, but well, I'll go ahead and mention this. African Americans respond differently to drugs than Caucasians. So there's certain things that you would want to give a African American, but again, it depends on if somebody's fully aware of giving everybody the same drug, it may not work in them. So yes, my wife deals with this daily in terms of blood pressures. She made the comment to me, if I can get them in a room and make sure they take their drugs and make sure what they eat, I can get their blood pressure down to normal. But again, it is not an easy thing to get somebody's blood pressure back to having an average, a mean of anywhere, say between 90 and 110. It's very challenging.
Yeah. Thanks.
Okay, you're welcome. So that's what we got. So one of the things we were looking at was trying to determine, can we figure out how, I guess I would say is how to figure out which one would have a fall in blood pressure. In these virtual patients, what's unique about them that we could do a test to determine whether they would have a fall in blood pressure or not. So if you think about this from a clinical trial, if the model is correct and everything is fine in that, if you could determine who to do this treatment to beforehand, you would save yourself a whole lot of effort. You would increase the efficacy of the trial. You could put it in the context of use in terms of who should use this. So basically, we want to do predictive testing and to leverage our observation about abnormalities within inside a network that can separate responding and non-responding phenotypes. So we looked at a couple of things that would affect renal function. One of them is a water challenge. You give them some water to drink. One of them is give epinephrine, which is a vasoconstrictor, which might affect salt and water. It does affect salt and water retention. Beta blockade is the other hand. Beta blocks epinephrine to sort of look at the response to thinking something else. So basically we came up with a potential predictive response, and we had a water challenge. And basically a person has to drink 250 mils of water over 2 hours, and we looked at how much they excreted over the next 2 hours. It turns out our analysis showed that the responders, those whose blood pressure fell with renal denervation, would excrete almost all of it. The non-responders would excrete only half of it. And it's a fairly tight range there. Now, if you give these people 1 liter, which is a substantial amount of water over 5 minutes, what happens is you actually affect other control systems, not just the kidney, but other hormonal control systems. And what you can see is you actually excrete over the next 2 hours. There's really not a whole lot of difference. They're fairly close with that. So this is one of those things that we thought about, and to be able to use potentially a clinical trial and a virtual clinical trial is something that we've been thinking about for a period of time. I'm going to talk about some other things in just a moment. So what we've been doing now is basically easy ability to generate large numbers of virtual patients. So we actually can sit on my laptop, and we got some Mathematica code to go through and analyze all the parameters. We do a sensitivity analysis to determine which ones to change. And we basically have code to be able to do it. And we actually can automatically call as many Amazon servers as you wish. Each server has 2 CPUs. So currently we're doing stuff with about 90 CPUs running at a time. And this was, when you talk about data, this was one of the things that we looked at, had last week. And basically this is the left heart pumping contractility. Contractility is usually between 1 and 1.2. And this is response to, they're hypertensive. And this is response to giving triple therapy, which is a diuretic, a beta blocker, and or maybe calcium channel blocker, and a angiotensin converting enzyme inhibitor or receptor blocker. So each of those time steps is on the bottom is actually a 12-hour time step. So you're looking at basically 100 days in the life of a person. And you can see some of them, the contractility comes back down to normal. Some of them, the contractility stays up. And the problem with the contractility staying up is you can end up getting a condition over a long period of time called heart failure with preserved ejection fraction. So that's what we can end up doing. I'm going to show you a few more things in just a moment, but we have a large model. It closely mimics real experimental data. We think it can be used to understand medical treatments. We can create virtual populations, and we're working to match experimental data. And the last thing is Jim Drew's actually working on this to basically be able to parameterize the model to match human clinical data. So can we look at, we're actually focusing on African-American populations. We're pulling some data out of our electronic data warehouse here at the medical center. And we're actually saying, can we look at something over a 3-year period of time, African-American population who are on multiple antihypertensives for that. So we do have some limitations. We don't have immunology, don't have genetics. We're hoping to get more data from human data from clinical trials or electronic health record, and ability to make multiple time-dependent measurements in humans is lacking. So I'm gonna stop there, but I'm gonna stop my sharing, but I'm gonna share something else just for somebody to get a good idea is, let me go share, and so Jim, I can help you with that. So this is what the model looks like if you run it on your desktop. Just to give you some idea, this particular version actually has 10,729 total variables and parameters. You can actually scroll through most of the list here. What I've done is I've loaded, if I remember correctly, I've loaded basically a steady state value, and there's the calculation in the 10 minutes in the life of the person. I can go through and look at all sorts of things. If I want to look at left heart pumping, systole, there gives you pressure volume, there gives you lots of other information. If I want to do respiration, look at gas exchange, they're telling me what happens with expired CO2, PO2, metabolism, go to different organs, then it gets sort of crazy. If I want to look at nephrons, there's everything you look at in the nephron, particularly glomerulus, and what happens with the filtrate, and there's what's in one kidney and the other, and there's again left, right, and left kidneys. Proximal tubule has two different sides, left and right. What can we do in lifestyle? We can actually schedule what the person does every hour of the day, what they eat. We can schedule what their goals are in terms of carbohydrates, fats, and proteins, and how many electrolytes. Environment, again, put them at Mount Everest. We can change the ambient temperature with that. We can do exercise. And we'll do that in just a moment. Let me get right for this one. We can diagnose lots of things from blood gases, things like chemistry chart, blood chemistries. Trauma is one of the things we're big on. These are things, blunt injury, hemorrhage, myocardial infarction. We have some more added later on. We can do sizing to change the organ mass and to change the, if I go to context, we can change the gender, age, height, adiposity, so forth. So what I'm gonna do is go ahead and put them on, put them at a slow level, put them on exercise bike, and have them go for, say, 5 minutes. And what you can see is there's what happens over the 5 minutes respiratory rate. There's what happens to our oxygen uptake. A little bit of an increase because of pressure at the kidney, left heart pumping. In-systolic pressure goes up a little bit. In-systolic volume goes down because they're actually putting out more output. Now, if I go through and bump it up again, and I forgot what his capability is to be able to exercise. And again, you can see that's gone up more. His left heart pumping has gone up. If I go back up to, sorry, there's so many things I have to, output that shows you his cardiac output. He's up to about 12.5 liters a minute. His heart rate is 126. We'll see how far we can push him in terms of this. 150 chart, go for another 5 minutes. He's still going. He's getting his heart rate up higher. His cardiac output has gone up more with that. So this is some of the things you can do. We can actually easily write scripts. They're a simple text file. It doesn't take very much to be able to do that. So I think I'll stop there. That's about 40 minutes after, and be glad to answer any questions or show anything else that might be of interest.
Thank you. Anybody?
Well, I'm just curious how hard it would be to, like, I'm doing some research with a colleague who's a mathematical modeler, biological modeler, and we're trying to build a simple, this is the problem. We've studied the Guyton diagrams and things, and we've seen some specific papers, but we're trying to adapt the idea to pregnancy, and specifically looking at preeclampsia, which is a fairly serious pregnancy complication. And we have actually dug into the literature from your university and from Guyton, and but we haven't been able to pull it in. It just seems like you guys live in a slightly different world somehow, in a good way perhaps, but it's harder for us to, you know, like, to understand and pull it out. Anyway, I just wondered, have you ever tried to think about pregnancy and how the model could be used for that?
It's interesting you brought that up, because we have 3 second-year graduate students that are doing taking our simulation course, and they just finished developing a very simple cardiovascular model. And we were talking about the 3 students about what they should work on. One of them is working really in a cardiac lab. One of them is working in an endothelium lab, and one of them is working with Barbara Alexander, who does fetal programming and uses a preeclamptic model with that. So we talked about maybe this person would be good trying to develop a pregnancy model, basically adding another organ that grows in size, which would be fairly straightforward. So I don't see that that's going to be a problem. the challenge with preeclampsia is there's a whole lot of inflammatory markers that are involved in that. And you could probably, if you collected data, particularly from animals, if you knew what was going on and look at the time response of those and then add those, that could be something. But drop me an email. We can probably could end up, I don't think that would be too, we have organs in there. So if you're adding a specific organ that has a metabolic rate and changes in size at a particular rate, I don't see that would be a terrible problem. And may I ask where you're located?
In Portland, Oregon.
Oh, yeah. Okay. Yeah.
And so we have, serious preeclampsia researchers involved in advising us. In fact, we have someone from Pitt who's helping us out as well. So we've got lots of good advice, but when it comes down to the modeling, and the problem is we're convinced that we want to build a model that is uber simplified compared to Guyton's model, but still useful somehow. And that's a big puzzle. You are saying the same thing, that keeping it simple is all relative, but it's an important part of the process.
Well, I think it's simple, but to be honest with you, I think it'd be relatively easy to add on with what we've already got.
Yeah.
Because there's a reason for that. I mean, ours, from the standpoint, it's already done, it's already built, it's got the hormones, the nerves, and everything else that you have with it. And if you look at preeclampsia, one of the consequences of preeclampsia is they get hypertension. And so we can easily go back and make for a variety of methods, we can make the model patient hypertensive.
Well, and they also get proteinuria, and that's
and we can add that.
You've got a lot of detail in the kidney already, so we should be able to do that as well.
Yeah, we should be able to do that.
Yeah. Okay. Well, I'll contact you offline. We don't need to take this whole group there.
Okay.
Thank you.
All right. You're quite welcome.
I just had one question, and that is, this is Mark Page. I have a little experience with this, and you've got about 2,000 parameters there.
Yes.
And you're going to have some limited clinical data, I imagine, to calibrate it. And I just wonder if you thought much about the strategy that you're going to use for calibrating to the actual virtual population.
So so one of these things I think we thought about is and I may be wrong about this, is can we generate a million patients and pick out those that might follow the pathway of an individual person? I mean, we've actually we've got an NSF grant, and that's part of it. We're going to have to generate about 750,000 virtual patients. So Drew Pruitt is the guy that's working on the parameterization. He and I actually had a chat this morning. He has a Ph.D. in mathematics. He is looking at doing that right now. We did something recently that basically we matched 4 parameters. Excuse me. We basically, I don't want to say cherry picked, but basically generated a virtual population, and then we compared it to a real population using the closest neighbor. And don't get me taught, I'm a physiologist, I'm not that. But basically, we could end up, on average, our virtual population matched a real population that was African Americans with high blood pressure who got calcium channel blockers, matched them at baseline on mean, and standard deviation matched them at baseline and at 3 years after cancer treatment with calcium channel blocker. So
That's hypertension.
That's for hypertension. Yes. So I mean, that was the reason the focus of the model was high blood pressure, and that's what we're really looking at, because there are people out there trying to figure out how to treat these patients, and especially it's a situation where they lack of care, and again, certain drugs don't work well in African Americans, and it may be the one that works really well on Caucasians, and you really got to figure out exactly what the best medication will work.
How many input parameters did you use to do that calibration?
I could not tell you. We actually tried to calibrate GFR and mean arterial pressure at the 2 time points. So again, we sort of got those things to match at 1 time point and matched at the end time point from a mean and standard deviation. But I didn't know. That was John Clemmer that was working on that. And I don't know which parameters he ended up using. If you send me something, drop me an email, I'll have John respond to you.
Okay. Just to let you know, I work at PwC, and we started a little spin-off venture called Body Logical that is attempting to do a lot of the same things you're doing here. There is a homepage for Body Logical, and you may want to take a look at that.
Would you drop me a email so I can sort of keep up with that? Yeah, we've got two other things that we do. The company does. One of them is fully running. There's actually a website called Just Physiology, which is basically for students. It's a browser-based version of HUMOD. And there's class lessons, and you can go in and you can put a person at altitude, or you can give them hypertension. And we actually have some patients there. I've got first-year medical students this week they're finishing the endocrine block, and they're looking at 4 different patients and providing their diagnosis. I've got a friend at University of Florida that uses it. He actually allows students to do research. So it's sort of interesting is, high altitude, what treatments can you give to let person survive longer at high altitude? I have a game I used with our first year medical students it's you're out in the woods with a friend and they start bleeding and you can't stop the bleeding. You've got 6 different treatments. How long can you keep them alive? And you've got to keep urinary output above 0.2 mils a minute as an index that the tissues are being perfused. So the kidneys are going to be perfused, they're making urine. So, it's usually, it's amazing you can actually keep the person alive for about 8 hours, losing about, I think it's about 10 mils a minute. And they get really low. And it's very interesting. And the model predicts, again, you would never get in a situation doing this is, one of the options you have is 100% oxygen. And you think, well, if you're bleeding, you're losing red cells and you're only putting back in just liquid, nothing that has red cells in it. But it turns out if you give 100% oxygen, there's a significant, it's not negligible portion of oxygen that can become dissolved in the plasma and transported throughout the body. So it actually becomes very important when you give them 100% oxygen with that. So
a lot of the relationships in Guyton model that as we understand it, are graphical curves that were experimentally determined.
yes.
Kind of varying one parameter and it's how it affects the other. How do you know that when those things combine, they combine in the way it really happens physiologically?
That's a very interesting point. And I think when Tom Coleman was working on that, I think we kept about 3,000 of his papers that he pulled out for all these things. One of the things we do is just sort of a proof of concept is we have a series of scripts that we run, sort of basic physiology. And we know what should happen in this case. And we run them and we look at them and we say, yeah, that's really what you would expect. If you're looking at quantitative versus, a 5 millimeter mercury fall in pressure versus a 10, yeah, we're not going to be able to pick that up. But it's one of those things that we just sort of routinely do that. And because we're trained to sort of look at the integrative aspects of it, we can say, yeah, that's what I would really expect. So it is a challenge with that. We're actually having, we've got a project right now and we're really struggling because there's not enough evidence about the effect of sympathetic nerves on a particular organ. And we're having to play with it a little bit to go back and see what's going to take to match up with clinical data. So it's sort of you try it out, does it match? Well, okay. And then you just keep adding complexity to it to basically how does it end up matching? And, we look at things that are simulated out 3 years. We do a 3-year simulation. It doesn't blow up. It doesn't get hypertensive if you have our normal patient. So yeah, we're always a little dubious about it working, but for some reason it works. And we think it works and think we got a good idea with that.
Some of those curves that we saw are almost straight up. So you don't have to move the X very far to really shoot the Y up. Yeah, it's kind of scary.
That's from the really old ones. Now, most of the ones now are not that steep. They're a whole lot flatter with that. I can tell you what, he's not in very good shape. He can get about 150 to 200 watts on the bicycle. So, but these are things that we would expect.
Hey, Mark Page, I'm really glad you're listening in because one of the things I'm naively interested in, not hardly knowing anything about physiology at this point, but excited about learning, starting with 14 organ systems, what they do, how they work, and so on. What are the issues? Mark, you and I had a conversation maybe 10 years ago, and I was asking you, why don't we see more applications of system dynamics in biomedicine? And part of your answer was, well, we don't have the data and the data available to make it. And on the project we did, that was certainly the problem. We had to go out and request the data and get it generated in order to do the work. So a naive thing I'm wondering about, take a look at, as I take a look at Humod, I've got it loaded and starting to play with it, do some exercise and so on. I wonder if we can generate plausible, simulated data that would represent various relationships that we think exists, and then build models based on simulated data that could tell us the value of actually going out and collecting the real data and at what frequency. Okay, so it could be a step forward to say, hey, we ought to take a look at this. Because in my experience, a question we had for our client was, has anybody ever measured the longitudinal hemoglobin data for an individual patient? And the answer was no. And I asked, why do you think that is? And they said, well, we don't really have to report that. Oh, okay. Well, why don't we take a look at that data, see what it looks like? And that's what I'm interested in, playing around with Humod to see if we could come up with types of issues that might be amenable to that approach. So I'm glad you're here, Mark.
Yeah, that one's got that one has such a long time delay on it. It's going to be hard, and probably also people worry about the cost of measuring your hematocrit. Sorry.
So Jim, you're thinking about some kind of a synthetic data experiment to see whether or not you could recover the parameters.
Yeah, see if you can generate time series that are plausible representations of what's actually happening. And if they are, build models based on that, and then show what the models could show, and then motivate the going through the cost of collecting the real data to see what other mistakes did we make.
Yep. One question I just had about this was, you showed simulations at very different timescales. Some of them were minutes, and some of them were multiple days or even years.
Yes.
And I'm just wondering, how did you handle that? I know that's always a problem because sometimes you don't want to do minute by minute simulations for 3 years.
It's actually built in. So you just tell it what I showed you in terms of the simulation. It's built in that if you do 1 minute, it shows you like every 10 seconds. You can set the display interval. So the step size for the calculations is determined internally, but the display is determined basically for that. What I showed you was that's built into. It's a very simple file. It says, oh, if I'm displaying for 60 minutes, let's just display. If I'm running for 60 minutes, let's display every 10 minutes. So we have some things that are on our just physiology site that we have it as a high resolution. So it's actually, if you want to look at 10 minutes, it actually may show you every 10 seconds to give you a little better, higher resolution. And really more smooth transients versus some of them, they're a little bit jagged just because there's a transition during that phase.
Okay.
We don't, especially on the server side stuff, we don't want to see every minute. When we're running 3 years of something, it's the data sets are just big enough to begin with, showing every hour. And sometimes we may end up showing a week. And when you're looking
But for the 3 years, you're still solving every 10 minutes.
Oh yeah. Oh, it's solving. The step size will vary internally. It's just more it's more the how often do you save the data? So if you do 60 minutes, you save the data every 10 minutes. Or you can say, oh, let me run 60 minutes, but I only want to see the beginning and the ending at time 0
Okay.
and time 60. Those are easy enough to set up.
Got it, okay.
This is super interesting, and I'm really looking forward to learning more. And I'll reach out to you later.
Okay.
Could you put, or could somebody put your email in the chat? I think you mentioned at the beginning of your presentation. You could just go to that slide, or you can
I'll
just
get
type
it.
it in the chat. Whatever's easiest.
I'm putting it in chat.
Thank you. I have to leave right on time because I have a meeting with the person I'm collaborating with on this project I told you about. So like it's
Okay.
perfect timing.
Okay.
Great,
So it's,
thank you.
yeah,
See you, Wayne.
I hate to say it, if you type up Robert Hester, usually in Google, I come up toward the top.
That sounds good.
I'm not sure whether it's good or bad.
Oh, it's probably good. It probably means you get a lot more spam though.
Well, I appreciate the time today. I was telling Jim when we had dinner tonight, over the last probably 6 months, I've gotten more requests to do these Zooms of people that I had never had heard of before and didn't know were out there. So we're always looking to do collaborations. The server base, the AWS stuff is proving to be very interesting because one server costs 7 cents an hour, which has 2 CPUs on it. You know, we might run, if each patient might run 3 hours. So, you know, it's 21 cents to get 3 years in the life of a patient. And if you're sort of looking at large data sets, it doesn't take long to get a whole lot of large data sets.
Mhm. I thank you all for making time.
Appreciate it.
I'd like to explore whether or not we could have a follow-up conversation on our next meeting on Monday, May 16. To that end, if you have questions or comments that you think of afterwards, could you send them to me? And one of the things I'll be doing, I have the user manual for HUMAD, I have the code installed, and there's some exercises in there. I'm just going to play with it, you know, see what that is like. We could talk about that at the very beginning level next week or next month. But and then Oscar and I have talked about maybe comparing some dehydration studies that were done with HUMAD that's similar to the work that she's done with System Dynamics to see what's the same, what's different, what some questions are. So please pass along some comments or things you'd like to discuss in a follow-up session. We can put something together for May 16th to do that. Yeah.
So let me follow up. It's
Sure.
very interesting. The whole reason we got into virtual populations was that we were looking at hemorrhage, and this was about 7 or 8 years ago. And there's actually a study in the 60s where they actually bled people and 750 mils. And if you look at the mean, yes, the pressure went down. And you start looking at the individual people, there were like 2 people that fainted after they lost about, had big pressure drops. And there were several people that had very minimal pressure drops. And we said, okay, how do we go about modeling this if you have different responses? And I forgot about that. The hemorrhage stuff was basically ended up pushing us to say, okay, if we're going to do a model, we can do a mean, but not everybody is an average person with that. The other thing to mention is, Jim, if you want to send out the user's guide to anybody, if you have issues with running it or installing it, I'll be glad to do a Zoom, not a problem. Walk your way through it in terms of what you have to do. There are 2 different versions of the executable. It's actually all of the mathematical equations are written in text file. They're in a version of XML. There's like 2,000 text files. So when it reads, it's easy enough for you to go in and change it, and then immediately within about 10 seconds, you got your new version up and running. So there are 2 executables. One of them is called Interactive Controller, and one of them is called HUMAD.exe. HUMAD.exe, you just run it, it loads the program up. If you want to use the HUMAD Interactive, and the difference is the Interactive had all the little tabs at the top. The other one, HUMAD.exe, you've got to click on individual things. So you can't just set up your tabs at the top and go back and forth between the 2. is a little bit more involved in terms of getting those set up. You got to know, sort of know your pass, where the model is. And it actually has a separate little executable that runs. It's called Model Solver. But I'll be glad to walk you through it, get online, whatever, and do it. But I tell you, if you download it, try humi.exe first to at least play with it. And then if you get one more detail, I can work with you to give you access to show you how to use the interactive controller.
Great, thank you. Yeah, let me know, anyone, if you want the user manual, I'll get it to you.
Sure. I don't know how much time we have. May I ask something very quick?
Oh, I'm fine. I'm free. I don't know how long. Yeah, sure. Go ahead.
This my guidance medical physiology textbook, it was a huge inspiration for me when I was building a system dynamics model for like body water regulation and normal physiology, but then I turned it into a game basically for the treatment of hyponatremia, which I presented here last month. Anyways, by then I think it was 10th edition, and right now I checked, I think the book has now, it's the 14th edition by Coleman and Hall, and I remember
Just by John Hall.
John Hall,
actually,
sorry.
Who's actually my chairman.
Okay, Hall and Hall, another Hall, two Halls.
It's his son,
That's
who's a
the same.
cardiologist.
Okay. So one thing so I don't know about the recent editions, but one thing we all noticed, so we had a conversation about this with Jim. The guidance book has some, it has the systems approach somewhat embedded and infused in it.
Correct.
It's also like a way of teaching medical students about physiology, but more in kind of in systems terms. To some extent, at least. So I was thinking by then it's a great start. And I was hoping that this would get bigger and bigger. I don't know if this is something that you would like to pursue, or like, I was just checking some of your publications before the presentation. I saw some papers on system simulation modeling for medical education, etc. Is this something you're pursuing?
Yeah, so we actually, so the just physiology is more for undergraduate students. I have this other project, which is sort of COVID sort of slowed it down. We're about ready to beta test it. My wife, as I said, my wife's a physician, and she has to deal with that nefarious electronic health record, which is made by Epic out of Wisconsin or wherever. We actually now have virtual patients that are embedded into Epic. So the whole idea was that students learn to document appropriately. And so you go into Epic and you have a patient, and it's basically HUMOD running on the back end that has a disease, more chronic diseases. You have to, as a history that goes with it, you can do lab values. You can then provide treatments and say, I'll see you in a month. So 30 days from today, you got to notice that your patient shows up. So it's more real time. It can be sped up, but it's more real time. We just sort of got sidetracked with COVID and all the clinical trial stuff. So it is there, it is up and running. We just haven't spent a whole lot of time on it in the last 2 years. So that's the other thing that we have.
Yeah. So another maybe more like high level question. So for us systems modelers with some background in physiology, or maybe less or more, I have a master's degree in physiology. So I'm familiar with or comfortable with the medical literature. So I assume you go to conferences mostly under like American Physiological Society, and I imagine there are a bunch of different journals that we can possibly aim and publish. And some of the conferences, do you know which ones of those are more open to modelers of integrative physiology? Yeah.
Well, there are two that we've published in. One of them is heart and circulatory physiology, and the other one is regulatory. Those are the two ones. Yeah, I think it sort of depends on which area you're looking at. Regulatory tends to carry most of the renal papers. Heart and circulation is circulation, but again, they'll take renal papers. There's a respiratory, there's an endocrine, there's a GI. I'm trying to think how many different sections there are, but mostly just because we're more cardiovascular renal, we've done both renal, regulatory, AJP regulatory, and AJP Heart in circulatory physiology.
But this academic society, like I'm asking if there are any threads particularly dedicated to physiological modeling, like or modeling of an integrated physiology for us modelers, like which of those conferences should we aim for, or maybe design it? I don't know.
Yeah, it's a little hard because I'm in the past was a member of the Microcirculatory Society, which was mainly microcirculation, and they have a strong modeling group, but they're looking at capillary flow through a tissue and red cell flow and oxygen flux and things like that.
Yeah.
Which it's fine, but I have a little problem about how do you validate it? You know, how much is going on? I'm trying to think. We've also published in PLOS One and also Frontiers. Frontiers has a really specific computer modeling, medical computer simulation section.
Thanks.
Just drop me let me think about that and drop me an
Yeah.
email so I get your contact information. I'll sort of think of other places.
And some of your recent work is also published in neuroscience behavioral reviews, neuroscience.
No, that's not me.
Oh, that's not you. Okay.
That's not me.
Okay.
I'm RL.
Oh, that's another. That's okay. Yeah, I was trying to understand the switch.
No,
Or is there a different research interest that you're combining?
No, it's not.
Okay.
Well, folks, it was great. Robert, thanks so much again for making time, just showing an interest and helping us understand. Very much appreciated.
Okay, no problem. Like I said, any questions, just drop me an email and can get online later on sometime. Bob, good seeing you again. We spoke